Hello, everybody. My name is Gary Schroth. I'm a Vice President and Distinguished Scientist in our Product Development Group at Illumina. And today I'm going to give you a short presentation on index hopping, some of the effects and mitigation strategies. So the purpose of this presentation is to provide insight on typical levels of index hopping that you might see and some of the downstream effect it might have on applications. We'll be covering what causes index hopping, reviewing some of our current mitigation strategies, and also discussing some future developments that we're working on inside Illumina. So outline of the presentation is as follows. We will give you an overview of index hopping, introduction, give you some insight into some of the mechanisms that we think are causing index hopping at a molecular level, and talk about some best practices and the differences between different applications when it comes to index hopping, and then also talk about our roadmap and mitigations for limiting index hopping. So what is index hopping? It's when reads from a given sample are incorrectly demultiplexed and mixed with a different sample or samples in a pool. Index hopping over the last few years has had multiple names: index swapping or jumping, index crosstalk, contamination, misassignment, sample contamination, or increased demultiplexing noise. These levels of index hopping can be mitigated through best practices that we'll be talking about throughout this presentation, and completely eliminated from your final data through the use of unique dual indexing, which we'll also be talking about. And one of the major conclusions that we've seen from looking at a majority of applications is that even though you can detect index hopping, typical index hopping levels do not really affect the outcomes of most experiments. So here's an example of one of the mechanisms that we believe is happening during an index hopping event. Essentially, index hopping leads to what you might think of as a molecular recombination event between the two ends of two different molecules. And so one of the biggest drivers, and you'll see this as a theme throughout the presentation, is that free adapters can cause a lot of these effects. So if you have free adapter, as shown on the right here, this free adapter could potentially bind to a strand inside of a cluster and basically be primed this way or that way to lead to sort of mispriming events or a hopping event. These strands can also float off and go seed other wells, and you can see that both the surface strand and the incoming strand could be potentially misprimed during these types of mechanisms. So this diagram gives an overview of the multiplexing and index hopping phenomenon. So let's say you're doing a simple library prep and you want to mix these two libraries together, and you've created them such that they have I5 and I7, which is our typical nomenclature for the indexes at the other end of our fragments of our library. You prepare them with a blue set of I5, I7, or a purple set of I5 and I7. And if you pull them together and then sequence them and go through demultiplexing, ideally all the blue ones would sort out together during normal multiplexing, demultiplexing, and all the purple ones will sort out together. And then when you look at the inserts, the green and orange segments here, you'll get nice pure inserts for doing your analysis, your alignment and analysis. But in the cases where you have a little bit of index hopping going on, some of the fragments will contain unmatched, unexpected pairs, where instead of having a blue-blue or a purple-purple, you'll get kind of a purple and blue kind of combination. And then what that leads to is a small amount of potentially contaminating reads in your expected reads. So as I'll talk about a lot during this presentation, unique dual indexing enables hop reads to be completely removed from the data, and that's one of our key mitigations for this phenomenon. So let me talk about some of those current mitigations. So we have, actually, before we get into that, I want to talk about some other factors that can increase levels of index hopping. So these are three things that we've looked at extensively, and in general, we're going to talk about library purity, library storage conditions and library prep methods, by far, as you'll see in some of the upcoming slides, the purity of the library is probably one of the most important factors that we've seen that can lead to very high levels of index hopping, much higher than expected. And so as we'll emphasize over and over, it's really important to clean up your libraries very well to manage this. We also have seen effects, though, of library storage, and so we will be giving you some guidelines and sort of updated guidelines in terms of library storage, in terms of trying to keep the libraries individually stored at -20 as long as possible, pooled just prior to sequencing, and of course don't store libraries at 4 degrees C or especially at room temperature. And all those recommendations are sort of within reason of your protocol. When it's possible, try to keep them at -20. And then as you'll see in some of the slides, library prep methods do have an impact on this, and PCR-free, for instance, shows much higher levels of hopping compared to our other library preps that I'll refer to as sort of PCR plus. One final note here is that loading concentrations, we've looked at sort of up and down of 2x of what we would want to load into a flow cell. It has very little effect on index hopping, much less effect than some of these other factors. So free adapters can induce high hopping rates, and this is actually a figure that is taken from our white paper, which I'll reference at the end of the slides, or at the end of the presentation. But what we did here is we took PCR-free library, spiked in with free adapter over a range of 0 to 35%, and you can see that this library started out with, say, 1.5% total index hopping, and as we spiked in the free adapter, you can see the total index hopping goes up pretty much linearly, get a linear dose response with the free adapter that's added. And so you can see here what we think is part of the mechanism. This is the structure of the PCR-free library, the control library here. And what we believe is that when we spike in the free adapter, which has these sort of forked adapter ends, as we called them, which is similar to what we have with PCR-free libraries, we think that adding those together is part of the mechanism, probably not the complete mechanism, but the point is that having free adapter at these kinds of levels of 50%, we can induce extremely high levels of index hopping. And in fact, most all of the cases that we've seen in the field that reported levels up in the 5% or greater, it has almost always been due to the fact that they had very, very high levels of adapters that are free in the prep. So this is an example of how library storage conditions can alter and increase the rates of index hopping. These are some experiments we did where we took libraries, these are actually nano, so these are PCR plus libraries, and we left them 1 week of storage at -20 degrees, 4 degrees, or at room temperature, and this is denatured after storage or denatured before storage. And what you can see is that actually this bar here actually goes all the way up to 7.1% index hopping. So storage of undenatured libraries at room temperature can lead to very high levels of index hopping. I doubt if anyone stores their libraries for a week at room temperature, but basically I think you can see the trend that keeping them at -20 degrees is by far the safest way to store the libraries. And so when I move on to current mitigations that we're working on, and one of the most important things that I've already mentioned is to clean up free adapters. That is by far, as I've mentioned, one of the biggest drivers. And then using unique dual indexing combinations. And I want to get into a little bit of detail exactly what I mean by unique dual indexing, because many of you are probably using dual indexing, but unique dual indexing is a special combination of dual indexes, and we'll talk about that here. So if you used our kits, the TruSeq kits, the high throughput or HT kits, they come with plate layouts. They're called DAP or DNA adapter plate, or RAP for the RNA kits, RNA adapter plates, and they essentially are combinatorial combinations of 8 different I5 indices on this axis and 12 different I7 indices on this axis over here. So we've got 8 I5s here, 12 I7s here. And if you look, at any given combination, you can see it appears to be unique, but just imagine you grab this column here. What you would see is you would have unique I5s on one side, but they would all share the same I7 on this side. And according to the mechanism I've been describing for index hopping, we wouldn't be able to necessarily ascertain if you had hopping or not if you use this kind of combination. But you can imagine, say, using a diagonal of combinations where they will all be unique. And instead of diagonal, we actually have come up with a little bit more sophisticated combination of this plate that causes a really nice set of unique dual indexes that has both unique combinations plus good combination to have color balancing and all four of the bases present in every read, et cetera, et cetera. So for instance, these purple boxes here, if you use those 8 combinations in a prep, then you would be assured to have 8 unique dual index sets. And so you can see each color combination here will give you a different set of 8. So if you kind of think of this combinatorial plate in this way, each plate then contains 12 sets of 8 unique dual indexes, which will allow you to sort of alleviate and eliminate this from your data, as I'll show in the coming slides. So one of the most important workflow mitigations is to try to design your experiment in a way that you can utilize unique dual indexing combinations. And as I said, today in our kits, we have 8 available. In our Nextera kits, if you're familiar with those kits and you use the same sort of logic, there are sets of 16, 24 different sets of 16 unique combinations that are available there. This is another way that sort of sums up those color coding patterns I was showing you. This is data taken, this table is taken out of our recent white paper as well. And this just explicitly tells you which well combinations to use to create the best unique sets of 8 using the RAP or DAP plate. So this is another experiment that we've done where we've taken several of our different flavors of preps, either PCR-free, TruSeq Nano, TruSeq stranded mRNA, the Nextera prep, or TruSight 170, which is one of our most recent enrichment preps. And this just compares the total level of index hopping from these various preps. And as I've referred to already, PCR-free tends to have the highest level and seems to be most sensitive to sort of levels of adapter contamination and cleanup problems compared to some of our PCR preps, which generally have very little free adapter in them at the end of the process. But PCR preps, PCR plus preps tend to have much lower levels in the range of 0.2 to 0.5 total compared to PCR free. So this shows the results of taking the unique combinations of adapters and using them in a 4-plex to create PCR-free libraries. So what we did is we made 4 libraries using these combinations, 702, 502, 705, 503, et cetera, that are shown in these green boxes I'm pointing to. And we sequenced those, and then we demultiplex them, and what we noticed is that using this kind of approach where we would not expect any of these red or pink boxes to actually have any library in them because we didn't use those indexes, because of this swapping or hopping phenomenon, we do get some signal showing up, and if you notice, it's basically in the row or column of each of the indexes that were used. So that's sort of a hallmark of this recombining effect that we've been talking about. And so one of the ways in all the previous slides when we talk about index hopping, essentially what we're doing is running an experiment like this and then essentially summing up all of these pink or red boxes, and that's what we call, in this case, 1.6%. You can see that the actual measured amount from any given library into another is actually much lower, but if you kind of add up all of this or add up the rows and columns for any given library prep or for any given library, you will get slightly higher levels depending on how you add them. But one important thing to keep in mind is that if you use unique dual indexing, essentially what you're doing is eliminating those red or pink boxes from the from the analysis. And so the unique dual combinations allows you to, as part of the demultiplexing process, essentially remove these unexpected combinations and filter them out, and they just become, essentially some part of the filtering noise of doing an NGS experiment. And so another way to look at it is like this. If you ran this experiment and you were using unique dual indexes, you would get, 26, 22, 23, 23% of the libraries would be assigned to your 4 samples very cleanly without any potential contamination at all. And you would get a pile of 4% that are unassigned reads. These would be, most of this pile is actually due to normal demultiplexing, errors in the demultiplexing read or other things that happen during demultiplexing where we would filter them out. And about 1.6% of this pile then would represent the index hopped reads. And if you were interested in studying exactly how many you could sort of make that matrix, that pink and green matrix we showed on the previous slide. But the main point is that just as part of the normal demultiplexing process on our system, you'll be able to put these hopped reads into this unassigned bin and only work with clean data. Okay, so let me talk now about some of our future developments. We've been studying this phenomenon for many months now. We've been working on specific new products to address some of these issues, especially as you know, we've launched the NovaSeq system earlier this year, which requires, it has enormous output, and for many applications it requires a high level of multiplexing and indexing, and so we are motivated to kind of get both better indexing and also get this, the purity of indexing to be as clean as possible. So we've been working on new adapter designs, a series of unique dual index kits, some enzymatic treatments that we'll talk about, and then we also announced about a week ago that we were going to we are in development for a multi-sample flow cell loader for the NovaSeq series. So let's talk about unique dual index combinations that we're working on. As I mentioned, in our current kits, the HT kits, the DAP and the RAP plates, you can find up to 8-plex combinations that are unique to do your work now. In the future, over the next few months, we'll be launching a 24-plex unique dual index kit of forked adapters and also a 96-plex. So these are the same kinds of adapters that work for PCR-free and all of our other TrueSeq style preps. And these will be unique index combinations that will ensure that any unexpected combinations can be removed from the data. So we think this will allow very, very clean plexing up to 96 plex per lane using any of our flow cells on any of our systems. And so this is something we're working on together with a strategic partner, and over the last 6 years, we have made 6+ years, I would say, we've made many different kits for many different applications, from whole genome sequencing to RNA-seq, and so we think we have a really good handle on how to make an optimized set. These will be highly purified, highly clean, and just ensure absolute 0 levels of index hopping and allow us to remove all levels of index hopping in our samples. The other thing that we're doing as part of these new adapters is we are building in specific modifications into the adapters. And so you can imagine that after the ligation process, these ends are exposed to solution, if you will, the 5' end of this strand and the 3' end of that strand. So what we do is we block in the duplex state of the adapters those ends, and so if they are not ligated together, we can use various treatments to fix that problem. We have used, for instance, a degradation, enzymatic degradation process to chew up free adapters so that these fully ligated adapters wouldn't be affected. We've also used other types of treatments as shown on the next slide here, enzymatic treatment that is essentially more of a blocking effect, where we block all the free 3' ends, which essentially stops a lot of this hopping from happening due to the free index. So a combination of both exonuclease treatment and a blocking chemistry we find actually also works really well in controlling this hopping, actually keeping levels low, even in cases where you don't necessarily have the opportunity to use unique dual indexes. So for instance, let me show you an experiment where we started with PCR-free library, generally got 0.8% essentially as the control. If you treat with one of these enzymatic protocols, you can knock it down even lower. If you add adapter, just like we've shown in sort of the spike-ins, if we spike in adapter to very high, we can induce very high levels of index hopping up in the 7% total hopping across all the combinations. And then by using this treatment, we can again knock this back down to essentially control levels with enzymatic treatment. And we actually have used this protocol with good effect on many different library preps now, where for whatever reason, either unique dual indexes were not available, or a specific cleanup was not possible to eliminate free adapters, which is rare, but that does happen in some preps. We can use this treatment to mitigate and knock it down to much lower levels. And the other thing to remember is that if you're using unique dual indexing, in the case of any of these preps, you would be able to filter out any of these levels of hopping using the unique dual indexing logic like we described a couple of slides ago. But there's no reason to filter out 7% of your data if you could instead just filter out 0.4 or 0.3% of your data. So I think that's another reason why these enzymatic treatment steps may become important in some preps. And finally, one of the other sort of significant developments that we're working on is specifically related to NovaSeq. So NovaSeq, as we said, is our most powerful sequencer yet, and it, especially if you imagine what we call the S4 flow cell. If you look here, you can see the number here, the S4 flow cell, which has four lanes apparently, but heretofore we actually weren't really offering individual addressable lanes. We were planning on having one loading port and that the instrument would then perform clustering of the sample through a single port. I think we got a lot of feedback after launch that was going to be enormous pools of samples required to do even RNA-seq and exome sort of library preps on these. So we took that feedback and we've decided to release this device that will allow individual addressable lanes on the NovaSeq. So that sort of reduces the sort of burden for indexing by a factor of 4. So instead of one sample pool going across all 4 of these lanes, you can put one sample pool per lane. And so that will change things quite a lot in terms of, I have some numbers here about using the 4 ports for, say, doing exomes or doing RNA-seq. And you can see that each lane then would have about 750 gigabases of data, or 2.5 billion 2 by 150 base pair reads. And so if you imagine doing exome sequencing, that's about nearly 8 gigabases of data per exome, or 26 million reads per sample for a 96-plex pool. So these are actually both really good numbers for doing exome sequencing or RNA-seq sequencing. So we think that the combination of the individual addressable lanes and the 96-plex library prep adapters that we will be coming out with later on this year will be a nice combination where you can run up to 384 different samples per flow cell in the S4 configuration and be able to explicitly remove any hopping that may have happened during your experiment. So the conclusions from this talk, hopefully you realize that probably the best thing to do with any of your preps is to use these best practices to ensure that you have very low levels of free adapter as possible. Even if you are using unique dual indexes and you can filter it out and you don't have to worry about it contaminating your data set, you still don't want to filter out more data than is possible or than is necessary. So, and whenever possible, use unique dual indexing strategies for sequencing. And then when unique dual indexing is not possible or when it is difficult to get rid of the free adapters from certain preps, then we are going to make these library treatment enzymatic reagents available over the next month or so to minimize the impact of free adapters in your standard preps. So finally, what I want to do is thank you for your attention and also point you to our website where we have a minimize index hopping page. If you are on our website and you look in the science area under education for researchers, you can find this page, minimize index hopping. You can download our white paper right here that I referred to several times during the presentation. Many of the data slides are taken from this white paper. And we'll be updating this as we get new information about index hopping and as we have new products available.